So today, I'd like to introduce our research on the esophageal cancer. Probably this cancer is seldomly occurrence in the other countries, in the Western countries especially, but the prevalence is still in China and also in the south parts of China, central part of China, and some part of the South Africa, this area. So I'd like to introduce our work on the etiology and mechanism study, and also we try to find some drugs can target this molecule targets. So because yesterday come here, many friends from Korea are curious where I come from. So I'd like to first introduce about our university. Our university here basically is at the central part of China, and we have university have over 70,000 students, the biggest university in China, and also our medical college is set up almost 100 years, and we have 13 affiliated hospitals, and our first affiliated hospital have the beds over 10,000 beds in single hospital, and totally these 13 hospitals we have over 40,000 beds, so also the biggest hospital system in China. So go to the topic. Today I'd like to introduce the Henan province here, actually at the central part, just I showed here. Here's Hebei. We see He is a river. Hebei is the north part of the river. The river is the yellow river. It's the very longest river here. So at this part, the red one, the deep red one, here have a high incidence of esophageal cancer. Here in the Uyghur region, and also have high incidence. In the south part, also have incidence of esophageal cancer. So that's esophageal cancer. Not today I start to study. Actually, my teacher's teacher, they at 1950s they already start to this esophageal cancer research. At that time, after the set up the country, the government very focused on the high incidence of different disease. Here's part in the Henan province, especially in the Taihang mountain area, the esophageal cancer incidence is pretty high. At that time, the government organized two medical research team and clinical doctors go to that area. So the pioneer, the professor is name is Sen Chung. Actually, he lived in the mountain area for do research almost 30 years. He together with the people there, and he actually invents a special screening, esophageal cancer screening system. That's kind of balloon, like balloon. He likes the patients, takes the balloon and then makes the air in, and then takes the balloon out. And the outside of this balloon can take some actually cancer cells through the cytopathology. He can detect these esophageal cancers. So at this age, they use this very simple methods. He is screening many early stage esophageal cancer patients and save many patients' life. So about the esophageal cancer research, many studies already show some risk factors like the neutrosamine, and that's actually it's already known it's a carcinogen. Also in that year, the epidemiology study found the fungus pollution, and also the lack of some certain trace elements like the selenium and also ions, and also like the vitamins like vitamin A, vitamin C, and also the fresh vegetable fruits intake is reduced. And also tobacco. Here I labeled with red color here, hot food and hot drinks. In that high incident area, people especially like take the hot soups, hot food, and even hot tea, everything is hot. So we especially suspect at that time, at the 1960s, the people already think that probably the hot drinks, hot food habits and cause esophageal cancer happens. But that's truly don't like in Korea, people like drink the water is mostly cold water, but in China people drink a lot of tea. That tea especially very high temperature, and also recent 20 years studies focus on the genetic reason, especially one paper published almost 10 years ago. One Chinese scientist found in the esophageal cancer the mutation of these genes like TP53, RB1, like CDKN2A. Actually, this has truly happened, but the esophageal cancer, not like the lung cancer, adenocarcinoma. That's the question I asked the other scientists several times, like why we found these mutations like EGFR in adenocarcinoma or other adeno cancers. Now it's mostly our target therapy is on adenocarcinoma. For the squamous cell carcinoma, mostly we don't have the target drugs. That's one very interesting phenomenon. The esophageal cancer is the same. Basically, it's a squamous cell carcinoma. See the P53 mutation is pretty high, more than 80%, and also other well-known genes related with the tumor genesis. But unfortunately, up to now, there is still no target drugs can treat this esophageal cancer. So in our study, and I just give us a recent samples of what we do. And one part we want to explaining how this hot drink or hot food habits can induce the esophageal cancer happens. And another story we told and how we try to use the samples and try to find the epigenetic factors, not the genetic mutation, but epigenetic factors. And then I introduce, according to these targets, how we try to find the repurpose of drugs for the cancer, this cancer prevention. So firstly, this just as I introduced, and the people like drink hot soup, hot tea, and also the hot food. That's interesting because we suppose this hot, drink food that probably cause of this esophageal cancer. So we make a model. We let this mice takes the hot water, treats the mice use the hot water like 65 degree, and together with one chemicals 4-NQ-O, and these models can specially induce the esophageal cancer happens, and together with the oral cancers. So at this stage, we found very interesting. We focus on the signal transduction pathway changes. Here, we measure all the stress-related pathway and the growth-related pathway. We try to find the interactions, like heart treatment is kind of stress, how this can stress, and the long-term treatments can induce, become a growth signal. Here we found a very, very interesting thing is we found the ERK, actually the treatments of hot water can cause the ERK pathway strongly induced. At the same time, we also found the P38 here is also strongly induced. We know that in the MAPK pathway, and the well-known P38 pathway is a stress pathway, mostly caused the inflammation factors. But the ERK pathway is well known for its related with growth. So how these HARTs at the same time induce the activation of this stress pathway and the growth pathway, we also test in the use the IHC. What we found actually this like the cell proliferation signals is strongly introduced, and also the phosphorylation of ERK pathway and the phosphorylation P38 pathway is correlatedly strongly induced. Because we know that both P38 and ERK, they are separated pathway, but at that time we supposed, we hypothesize whether this P38 pathway can directly activate the pathway. Basically, that's different with the traditional series, but we found actually if you use the P38 as a kinase and you phosphorylation ERK, we do this in vitro experiment. We found actually the P38 cannot directly phosphorylate the ERK. So we suppose probably, this kind of stress pathway can interact with the ERK pathway in an indirectly manner. So here we use the P38, we do the put-down assay, and then we do the MS. And very interesting, we found one target, ACLS4. This actually is enzyme. The enzyme actually is kind of messy. It's catalyze the methyltonic acid and to the acetyl CoA. This kind of catalyze, can the substrates and is methyltonic CoA actually is the substrate for the SAC phosphorylation activation. Here, interestingly, we found the P38 through the iPSC. We found they strongly work together with the P38, and interestingly, we found that P38 here can phosphorylation that ACLS4, and later we identified this new phosphorylation site is thrombin 679. It's a new phosphorylation site. We make the antibody through the commercial company, and we verify this antibody, and truly through in vivo and in vitro, and this is through mutation, and can strongly active this phosphorylation site. So we check whether ACLS4 is also working, have a function in the cell proliferation and related with the patient's prognosis. And we found here actually truly that's the ACLS4 and also highly expressed in the esophageal cancer patients and also related with the patient's survival. So next question, we try to find this for high level expression and phosphorylation whether it has a function or not. So here we knock down ACLS4 in different kind of esophageal cells, and we see the clone information and the proliferation is also repressed. And interestingly here, we make the mutation, the function reduced mutation and function, the A mutation and the D mutation, B mutation. Basically, the active is phosphorylation style mutation, and A mutation is kind of function lost mutation. Here we found actually this active mutation have the functions to stimulate the cell get more cloning, and the A mutation actually suppress the function. So next we check whether this kind of phosphorylation, the products of methyltonic CoA's level, we found actually this knockdown ACLS4, the level is decreased here, and we also use active mutation and function lost mutation. We found this D mutation is active mutation. The products is storic COA level is increased after the function loss mutation, the level is strongly suppressed. So because it's a substrate of the SAC phosphorylation, we check the SAC phosphorylation. Here we found the SAC phosphorylation actually with the demutation, the SAC phosphorylation strongly increased. Here we use the originally introduced model. We check these signals. We found this p38 phosphorylation, the ERK phosphorylation, and also the phosphorylation of ACLS4 is strongly correlated, and also the products is increased with the active this mutation. Then we also make a knockout model here, and with the model we knock out the ACLS4 and introduced with foreign cure O. Here we found very interestingly, with these models we see here the p38 phosphorylation, foreign cure O mutation, conditional knockout esophageal cancer. We see the phosphorylation of p38, the ERK, and also the SAC, especially ERK and SAC phosphorylation is strongly reduced. And this work published this year on the Nature Communications. So interestingly here, the major contribution I think is explaining how environmental stress and active this stress pathway, and through the metabolism disturb, and especially the lipid metabolism disable, and can active the cell growth pathway and cause the cell continued growth. So, as I said, we also very interested in the molecular pathway of esophageal cancer, and we tried to find some not genetic. We know the genetic mutation didn't work, didn't find useful targets. So we tried to find some epigenetic factors whether can be targetable and for this disease treatment. One story I told today is we found that the BRD4 is kind of broad domain, and the epigenetic regulators and can regulate the RCC2 is strongly related with the tumor inflammation and tumor genetics of esophageal cancer. At this story, we firstly found actually the BRD4 at different panels, and we found that BRD4 is strongly overexpressed, and these different stages, and also related with stages. And here is the IgG staining. We found that in the esophageal cancer patient sample, the BRD4 is also strongly overexpressed. So we try to planning what's the meaning of the BRD4 overexpression. Here we use the cell lines we found at the esophageal cancer. Here's control. It's kind of epithelial cells, immortalized. Here we see different cell lines all overexpressed. So we knock out these cell lines, and we found so strongly surprised the esophageal cancer development, and also with the CDX model, and we see the cancer growth is suppressed. So we try to explain how BRD4 work on the esophageal cancer growth. So we use the inhibitors and also SH cell lines and do the sequencing, and we want to find the cross downstream targets. And interestingly, we found 11 interesting targets. At these targets, actually, we found one gene is very interestingly, it's RCC2. These genes we found after knockdown BRD4, and also use the inhibitors, the RCC2, the molecule is strongly inhibited. So because it's an epigenetic factor, we try to figure out how it's regulation, because BRD4 is just a broad domain recognized molecules. We try to find what's the transcription factors. Here we found actually the TP73, and after knockdown the P53, we see the P53 can the RCC2 RCC2 at the promoter region, the TP53 can bind with the RCT2 promoter region and together with the BRD4 and regulate its expression. So we use the PDM, we set up many PDX models that use patient samples, and on the mice we found very interestingly that if we, the tumor volume, if we treat with inhibitor of BRD4, either the KIR1 or OX13, the tumor growth is strongly suppressed. So this study that illuminated BRD4 actually is a very interesting targets in the esophageal cancer, can together with the TP73 and regulate the expression of RCC2, and this work published on the oncogene. And lastly, we always, when I was studying in the United States, our tutor, together with Professor Lee, we do the chemo prevention job, who after I go back to China, I continually for chemo prevention research, especially on this esophageal cancer. The chemo prevention, just as simply it's use chemicals or the healthy food and prevention the cancer happens. But I think that's not a new concept, because like 50 years ago or 70 years ago, and the patients actually they get like high blood pressure and high lipid cholesterol, and basically clinically they have no symptoms, but still at that time there's no drug treatment. But currently, and if you go to clinical do the regularly checking, you found these levels, glucose, these levels increase, and the doctor will suggest you take some medicines and decrease this glucose level, lipid level, and that's for your health. So that's just the cancer prevention. The symbol is also the same conception. Use these kind of chemicals can inhibit the cancer progression and actually extend the cancer occurrence process. So here we found very interesting targets. ITGAV is kind of membrane proteins. It's an integrin. Basically, it's a sub-unit of integrin type 1 and this alpha units and integrin 5, and is very highly expressed in the esophageal cancer. Here we saw in the different panels, and this prognosis, and all related and highly expressed. So we knocked down these molecules, and we found strongly also suppress the cancer development that's been and probably can function as a target. So interestingly, we use a computer model to the docking with screening the clinical already available drugs. We found a very interesting target is the indomethacin. Indomethacin is a very cheap drugs for the anti-inflammation drug. Here we found the indomethacin actually can interact with the ITGV and through the SPR and the other methods, the put-down methods. We found actually the ITGV basically can interact, indomethacin basically can interact with ITGV. And after the interaction of indomethacin with ITGV, we found a very interesting phenomenon. Here actually treated with indomethacin, And we found actually one ITGV, if to the ubiquitination, we found actually with these molecules, indomethacin, after interaction with the ITGV, ubiquitin can cause degradation of this ITGV. So we use all other different experiments that show that another ubiquitin enzyme, E3-like SYV1, can degradation ITGV, and also these procedures, if you added into medicine, can accelerate these protein degradation processes. Interestingly, here we do you also use animal models. We show actually this indomethacin can strongly suppress the tumor growth in different PDX models. And actually indomethacin effects it's stronger than the, this also go to the IRDG1 is already go to the clinical trial, but the indomethacin, we see the effects actually in the phase is better than it's already clinical available the drug, the clinical trial drugs. So, interestingly, because integrates strongly with the TGF-beta pathway, so we try to figure out whether it can suppressing the tumor growth, whether it can affect the immune system or the tumor immune system conditions. So here we found a very interesting thing. Just use the indomethacin treatments can reduce the tumor weight, and if you use the PBMC, use the immune cells together treats the tumor cells will strongly inhibit. So that means this indomethacin treatment also can cause the immune system changes. Here we check the TGF-beta levels. We found that PBMC plus indomethacin strongly reduce the serum TGF-beta pathway. And this work published on the Clinical and Translational Medicine by Dr. Fangfang Liu, and also she also do a lot of work still on indomethacin, because he strongly suspect this is a molecule glue to target this ITGV. So lastly, I'd like to thank Professor Dong and both Professor Li and we together work with Professor Dong still, and also my team, the other members. Thank you very much.